Good afternoon, everybody. Welcome back to the workshop. I hope you all enjoyed morning sessions and had a good lunch. We will start our afternoon session. The first one is IVIVC for complex parenteral and ophthalmic products. We'll have 3 speakers for this session. Our first speaker for the session is Dr. Xavier Papin from Formulations Plus Inc. He will be presenting on challenges and opportunities for developing IVRT and IVIVC of complex injectable formulations. Please welcome Dr. Papin.
Thank you, Jan, for the nice introduction. Can you guys see my screen?
Yes, it's coming up. Perfect.
So this is the outline of my presentation. I'll present. I've got an introduction slide on subcutaneous physiologically based modeling and why we need it for subcut modeling. And then I'll move to case studies. I'll speak about Exenatide formulations, which cover the range of simple solutions to extended release microspheres. I'll speak to the challenges with in vitro release testing method development and the opportunities we have in using PBPK or PBBM. I'll present the current advance on mechanistic release for PLGA microspheres, and then I'll finish with some take-home messages. So in terms of mechanism leading to subcutaneous absorption, there are quite many. Here I'm showing a scheme that I've developed for an IR suspension. Of course, depending on the mechanism of release, this could be more complicated. But as you can see here, there are many phenomena that are happening in sequence and in parallel, which can lead to drug losses or drug delay, which will eventually affect the rate and extent of absorption in the systemic circulation for a subcutaneous formulation. And if you think that this picture is complex, then you should think that all of these phenomena will change with time. So for example, the volume of the formulation in the tissue will change with time. The composition of the formulation will change because the buffers you've put in the formulation will exchange with the physiological buffers coming from the capillaries in the tissue. So there is a dynamic aspect to all these volumes and all these boxes you see here on the screen, which will have to be incorporated as well to bring more mechanistic and more physiological model. So because of all these parameters, we definitely need a PBBM, so physiologically based biopharmaceutics model, to try to understand which of these parameters is limiting, because we may not always be in flip-flop kinetics, and therefore we need to evaluate the impact of these parameters in sequence and in parallel. So moving on to my first example on Exenatide. So Exenatide is a 4 kilodalton peptide. It's hydrophilic and it's present as 2 formulations, one IR formulation called Byetta and 2 MR formulations, Bydureon and Bydureon B-size. The difference between the two is the solvent that is used. Bydureon is aqueous-based and Bydureon B-size is migliol-based. So I'll be speaking more on Bydureon, so the aqueous-based suspension. So in terms of PK parameters, we can access them. There's a study called 111 in the submission file, which is a 6-hour infusion of Exenatide in healthy volunteers. So if you feed that data, you can actually extract the disposition and clearance, which you can assign to renal clearance because of the mechanism of clearance for that drug. And when you look at the comparison between intravenous data and subcutaneous for the IR formulation, so the IR solution Byetta, the immediate thing that you can see is that the AUC for Byetta is dose proportional, but it's not exactly the same proportionality as the intravenous. So there is a lower bioavailability for those solutions. So it's not 100%. if you believe this data, you can calculate about 72% bioavailability from the subcutaneous injections for the IR solution. When you run PBPK modeling with GastroPlus, for example, using a subcutaneous solution and the base characteristics of physical properties of the drug, you do get to this picture here. So you do have a relatively good prediction, apart from some studies where, especially in study 102, very high doses, you can see you start to over-predict the Cmax dramatically. If you look at it in terms of observed versus predicted, the AUC is slightly over-predicted, about 20%. The Cmax is 60% over-predicted. And this is especially true for the higher doses. And then the Tmax is in the right ballpark, but with 40% under prediction. So it's a bit too fast in terms of prediction, and it's a bit over-predicted. So you could think that there are different mechanisms which could explain this over-prediction. Of course, in the model is initial base model, there's 100% absorption and 100% bioavailability. So the different reasons why we don't see 100% bioavailability for an IR solution could be chemical degradation, it could be metabolism in situ, it could be binding to cell surfaces or to the extracellular matrix, it could be physical degradation, oligomerization, and slow diffusion of these oligomers. So we're going to explore these reasons one after the other. In terms of degradation for Exenatide, it's been reported in the literature and it is pH dependent. It does chemically degrade and it also forms aggregate so there's a physical and a chemical degradation, which has been evidenced by size exclusion chromatography and reverse phase HPLC. And overall, the rates are more or less the same. They go down with pH and they are over the half-life of degradation is over 100 hours. So it's unlikely to be the explanation for the lower bioavailability for the IR solutions. Moving on, it could be metabolism for that drug, but here again, we have very short absorption time, roughly Tmax in 2 hours. The drug is not a major substrate to peptidase present in the subcutaneous space. And you could think that you could impose a first pass degradation with constant in situ clearance to simulate the impact of local degradation. But if you do this, for example, here I've put a very low degradation in situ, low clearance, you can normalize the AUC. So you go back to average fold error around 0.9, which is quite good over the dose range covered. However, the Cmax is still over predicted and the Tmax is now under predicted, and the under prediction is actually even worse. We have 50% under prediction. So the local clearance doesn't really help in our case to predict the whole profile. So what could go wrong? We ruled out chemical degradation and metabolism for the IR, but what about physical degradation or binding? If you look in the literature again, you can see that Exenatide is reported to bind to liposomes as a monomer and as a dimer. And if you look at the study, the high dose study, cohorts in the study, that was really mismatched compared to our baseline prediction, which was a 0.4 microgram per kg injection. You can calculate roughly the surface area that you can cover with that formulation, the cell, the adipocyte surface area. Roughly, the diameter of adipocyte can range from 100 to 123 micrometers, depending on the obesity of the subject. The RCS is the relative concentration of the solution in the depot, because the depot will form a tree-like shape in the tissue going around the adipocytes. And phi cell is the fractional cell volume in the tissue. So that gives you quite a large surface area. And if you just compute, the monomer size or the dimer size, you can actually bind from 6% to the entirety of your drug onto the cells. So there's definitely an element of potential binding, but it's not going to be 100% for sure. But there is this affinity that has been reported. The other parameter to consider is the oligomerization. So again, from the literature, we know that exenatide can form dimers in solution depending on the concentration. And the dissociation constant was actually measured and reported. So from that dissociation constant, you can solve this equation to find out the fraction of drug in form of dimers in the solution as a function of drug concentration. And for the Byetta concentration, which is 250 microgram per ml, you've got about half of the drug in the form of dimers. And if you look at the size difference between the monomer and the dimer, about 17% of the monomer is expected to be clear by the lymph. But if you talk about the dimer, this goes up to 32%. So there is an element of diffusion, which will be slower for the dimer, but also source of clearance. And when I see clearance, I mean absorption pathway, which will be driven by the lymph convection towards the lymph system. So when you put this all together in the simulation, if you move from a subcutaneous solution to subcutaneous suspension, if you put a radius now for your suspension, if you assume binding, if you reduce the diffusion coefficient, you still don't have any metabolism, but you can also reduce the solubility for the monomer because it will have to release from the dimer. You can start to explain some of the results that were observed for the solution for the very high dose in study 102, and you can have that match of Cmax and you still have a good AUC, but you match the Cmax much better than initially with the solution. Moving on to the extended release formulation. So Bydureon is presented in PLGA microspheres, and the mechanism of release is following an initial burst release. You have two phases. One phase, phase number 2 here on the graph, is the hydration phase, where the molecular weight of the PLGA is going to be reduced through hydrolysis. And once you reach a critical molecular weight for the polymer, the drug starts to diffuse out of the microspheres and release in the medium. When you look at the PK, you can very well see the burst phase. So you have that first 24 hours where the drug is released immediately from the microspheres. And then following this 24 hours, you've got a prolonged release for the drug, which is compatible with the formulation mechanism of release. And the dose proportionality is okay, but as you will see, it's much lower in terms of AUC compared to the IR formulation. So the burst itself is the loosely bound drug, and when you look at the AUC of that burst, and you calculate the percent AUC to the total AUC that you observe during the PK, it's a very small amount of drug. It's only 2% of, or less than 2% of the total AUC, and it is dose proportional as well. When you look at that study where volunteers were given 2.5 to 10 mg single dose. The IVIVC that was reported in the Clean Farm review on the FDA website shows a good correlation, I would say, between cumulated AUC in vivo and percent released in vitro. And you have that profile here, which still shows that the in vitro release method, which is the diamonds here in the graph, do not show any release over the first 20 days, whereas in vivo you have already drug appearing in the systemic circulation. So there's that mismatch here, which is apparent for the initial 20 days. For the burst release, it's treated with a different in vitro release testing. This is the complete release profile, which is looking at the extended release for the microspheres. Another important thing to consider is that this test is run at 37 degrees for the in vitro, whereas in vivo, the subcutaneous tissue temperature is around 34, depending, of course, on your activity and the outside temperature. But the baseline temperature is around 34 degrees in subcutaneous space. This graph shows you the relative bioavailability, the AUC, the absolute AUC of all those formulations. And as you can see very clearly here, even though we have dose proportionality for the extended release formulation, the AUC is not at all in the same ballpark as the IR subcutaneous formulation. So we are losing AUC, we're losing drug. The bioavailability goes down, and it's estimated based on the intravenous clearance is estimated to be between 9%, 10% to 15%, depending on the dose here and the AUC that were reported in the submission. So when you look at a cumulative AUC, it becomes misleading because not all the drug is bioavailable, not all the drug is absorbed or even released. So you are going to normalize with an incomplete in vivo release, and that can distort a bit the in vivo cumulated profile. That's an issue. There's a workaround, and you can use GastroPlus, for example, or as a PBPK platform to do mechanistic modeling. And here we use a top-down approach. So we use the measured PK profile for the 4 doses of Bydureon given as a single dose. And we fitted it with a triple phase viable function just to match the in vivo profile. So this will extract the in vivo release from the measured PK. The hypothesis we made is that we had similar release, so we're not changing the release according to the dose, but we are changing the local clearance. And this is consistent with the fact that we have now released over several days or several weeks, which may lead to local degradation. We've seen that the degradation half-lives were in around 100 hours at pH 7.4. So we are expecting some local degradation, and this has been demonstrated as well. That local degradation can be inside the formulation, or it could be outside the formulation once it is released. This local clearance is actually dose dependent. So when you do that top-down exercise, you fit the local clearance to match the PK. You can see that it increases with dose, and the reason why is probably related to the volume effect, because the dose were achieved by injecting higher volumes of formulation, and we know that the volume will occupy more space in the tissue. You'll have more contact of the formulation and the drug with potentially enzymes or potentially sources of degradation. We also know that the drug undergoes self-catalysis for the degradation, so the volume present and the dose present can also be a factor to speed up the local release. The other explanation to that degradation here, the hypothesis we made was that there was a local clearance. It could also be incomplete release because of the release mechanism. We have that gelling, but that gel remains for long periods of time, which could attract some macrophage locally, and you'd have other sources of degradation and not necessarily due to local clearance. But when you apply this top-down approach, you can calculate an in vivo release for Bydureon, which is here dose dependent. And if you plot it against in vitro data, you can see that mismatch already, and that was shown with the previous slide from the submission. So the first 20 days are showing very little in vitro release, whereas in vivo it has already started. And then the two curves cross at one point, and the in vitro will be then faster or more earlier, I would say, than in vivo. And the reason why I say happen earlier in vitro is because of that temperature difference. We know from the literature that temperature is very, the PLGA degradation is very sensitive to temperature. So if you increase the in vitro release method temperature to 37 and in vivo it's 34, this small difference is likely to induce a faster degradation of your polymer and therefore an earlier release of the drug. So one way of checking this is just to time shift. And if you time shift the in vitro release profile by 300 hours, for example, you can still see the mismatch initially with the absence of in vitro release, whereas in vivo you have some drug appearing in the systemic circulation. But later on, during the release period, the agreement between the rate of release in vitro and the rate of release in vivo is correct. And so that mismatch remains. I mean, however you shift it, you'll still have that discrepancy between in vivo absorption and in vitro, in vitro release. And that could be due to other phenomena happening in vivo. For example, it's been reported that people massage the injection point in vivo after application of Bydureon, so that could increase the stress, increase the local blood flow, the local temperature, and start the release a bit prematurely compared to what happens in vitro. So in terms of challenges for developing accelerated method, we started that discussion this morning, but there are different methods to accelerate in vitro release. You can play on the temperature, which will change the, if you move closer to the TG of your polymer, that will speed up the diffusion coefficient, will plasticize a little bit. You shouldn't go beyond the TG because you may change the release mechanism. And the other factors you can play on, if you have a pH-dependent solubility for your drug, or if you can use a solvent, you know that, and if the rate controlling the mechanism of release is controlled by the solubility of your drug, then that would be, you know, pH, and the use of pH and solvent would be as well a way to increase to accelerate the in vitro release method. And as we discussed this morning, you need to make sure you don't change the release mechanism compared to the real-time testing, because you may start to see things that are not predictive of what will happen in vivo. And I know we spoke this morning about the correlation between in vitro release testing and whether it should be biorelevant or biopredictive or not. Ultimately, we're looking at product quality, but we're also looking at product quality in view of what we give to patients. So if you completely change the release mechanism, if you move from diffusion to erosion, you may not be looking at the right parameters or the quality attributes of that you will see potentially with that release testing method may not apply in vivo. So the term quality, you know, you could still look at batch to batch variation, but what is the impact in vivo, and can we ensure our patients will get the right treatment? You know, this is the key thing behind not moving too far away from the biopredictive nature of the method. Of course, you need to correlate both methods, as we discussed again this morning. So you can define time scaling and release scaling with multiple variants. And this is also useful when you want to engage into safe space evaluation. So really recommend to develop those variants by changing, for example, the intrinsic viscosity of your polymer, the ratio of drug to polymer, the loading, the size of your particles, and verify that you have a discrimination. And if you have a discrimination in vitro, does it translate to a difference in vivo? So having those studies performed is really key, because they will inform the formulation scientist into the critical biopharmaceutical attribute, the CBA, which are the CMA and the CPP, so the critical material attributes and critical process parameters, which are likely to impact the PK. And of course, it's important when you develop an IVR method, whether it is accelerated or not, to understand the difference between your in vitro conditions and your in vivo conditions, whether it is the degradation inside the formulation or outside the formulation, potential adsorption to to cells, to extracellular matrix, the buffer nature and capacity, because again, these will exchange very rapidly after injection. The sink, we spoke about the sink level. In vivo, the fluids will not exchange so much. You will not have the same volume, the same sink conditions, and the also an important aspect in establishing IVIVC is to look at the bioanalytical method and the in vitro analytical method. They should really look at the same thing. If you have an ELISA method in vivo, and if you have a size exclusion method in vitro, you may not be looking at the same thing. You may have degradation that you don't see, or you may count drug that is degraded, whereas in vivo you will not count it. So that will impair your understanding and will impair the correlation, definitely. But there is an opportunity to a simple correlation, is to rely on mechanistic deconvolution of your clinical PK with PBBM, especially if you've informed the PBBM with other parameters such as the size of the drug, the diffusion coefficient, the degradation rate, because then if you extract that in vivo dissolution, that can serve as a blueprint to define your predictive method. So to finish on Exenatide extended release, there was an IVIVC study conducted in the file, and it failed on both AUC and Cmax, and the fast and slow formulations were actually less bioavailable than the reference list drug. And as you can see here, the relative bioavailability of Bydureon to Byetta varied during that study from 11% to 25%. So there was a twofold difference in AUC that was mediated by those variants. And there's really at the moment, no understanding of what happened here. But this is the thing you could be looking at when you do a study like this, with potential phenomena happening inside the formulation or outside the formulation, which reduce your AUC. So the conclusions on modeling peptide absorption is even IR solutions can be complex. So you have to look at potential binding, oligomerization, which will change the absorption pathway and may change the extent of, or not the extent, but the rate of absorption. For the MR formulation or ER formulation, the degradation rates of the drug should be considered, and they can be integrated in the in the model. And the model can integrate external measurements to the in vitro release, which would apply to more complex mechanisms. For example, if you have complexation or degradation following the release. There are still things that are really hard to model, but which can change either the volume around the tissue, around the formulation, is the immune response and inflammation that can happen following a long-term injectable, long-acting injectable injection. And there are different stages, and I'm not going to all the details here, but there are lots of different stages following injection. So just finishing on the future, this slide is showing the factors that can impact the drug release from PLGA matrices. And I'm not going to go through all the details. You'll have the slides and the hands out, but there are many different parameters. And within Simulation Plus, we've developed an advanced mechanistic model for the release of polymer PLGA microspheres that contain the acid degradation, the pH-dependent solubility of the API, and the diffusion of the water and reaction with a drug to mediate hydrolysis. And so the interesting thing about this model is that you can start to see differences depending on the size of the microsphere. For a very large size, a large particle, the acid which comes from the PLGA degradation builds up inside the sphere, and actually the degradation starts from the inside. Whereas for a smaller particle, the diffusion of the acid is faster, and there's no buildup of acid, and so it degrades from the outside towards the inside. And with this in mind, there was an attempt to predict the dissolution of piroxicam PLGA microspheres with 10 micron size on the left here and 50 micron size on the right. And although the agreement is not perfect, you can see for different intrinsic viscosities, there is a qualitatively good match between the measured in vitro release and the predicted in vitro release, which makes this tool very interesting because it becomes mechanistic, and you can think that if you can model in vitro, you can then use the same dissolution method in vivo with, of course, different volumes, different buffers, different exchange rates, and that would provide for a translation between your in vitro methodology and your in vivo release profile. So just to finish my presentation with some take-home messages, it's really important to integrate mechanistic in vivo release in PBPK and or PBBM. It's not necessarily always available, but we're working towards offering this possibility in the future because it will be able to integrate complex phenomena happening in parallel or in series. And it will definitely surpass the classical approaches because you can now start to capture, as I've just shown, the effect of particle size, of polymer viscosity, polymer degradation, temperature, the exchange of buffers locally. Of course, this needs more effort. You need to compute more things. You need to understand the effective depot size, the fluid exchanges, the local degradation, the binding. So you're going to have to measure more things outside of your in vitro release. You will need to measure these parameters and model them in your in silico tool. The other take-home message is be careful of cumulative AUC because it actually masks the actual AUC. If you haven't released or if you haven't absorbed 100% of your drug, or if it's not consistent across different formulation, you may actually distort the profile versus time when you do this normalization. And that is an issue when you want to draw correlations. And finally, I'll finish with some advantage of using mechanistic models is if you have a full PBPK model, you can then choose the matrix of the relevant matrix and relevant analyte to build your correlation. And I'll give you an example a bit outside of the examples I've developed here. But if you've got a locally active drug, you can use a downstream metabolite. If your drug, for example, is minimally absorbed, but it is transformed into a downstream metabolite, you can look at the pK of the metabolite, link it to your in vitro performance of your variants, and then recalculate the active drug concentration at the site of action. So it offers you much more flexibility to build correlation than just looking at the plasma if that's not where your drug is effective. And I would like to thank the three colleagues here for their help in putting up those slides and reviewing them. Thank you very much.
Thank you very much, Dr. Papin, for the wonderful presentation. Our second speaker for the session is Dr. Amy Barton, who is the scientific lead on nanomedicine at Vivo Pharma, Inc. Please welcome Dr. Barton.
Thank you so much, Dr. Wang. Can you just confirm my slides are visible?
Yes, perfect.
Perfect. Today, I'd like to discuss the inherent challenges with IVRT that are really unique to the IV iron nanomedicine complexes. And then I'll also describe how this could result in a potential bottleneck into development of an IVIVC. As a point of disclaimer or disclosure, I am an employee of V4. However, the views and opinions in this presentation are my own. I have 5 principal objectives I'd like to talk about today. One is to give a brief overview of the complex class of IV iron carbohydrate nanomedicines. I'd like to then compare and contrast analytes that could be potentially exploited for IVRT. Then I'll discuss how these could be applied to IVIVC models and relative caveats. I'll be reviewing data that were gleaned under an FDA U01, for which I was funded while I was still an academician at the University of Michigan. And then I'll sort of dovetail all of these data into a final discussion on the remaining research gaps around this complex class of parenteral injectables. The need for iron supplementation has been known for centuries. However, early studies that attempted to administer quote unquote naked polynuclear iron oxyhydroxide were associated with serious adverse events, which in fact in 1946 led this group of authors to conclude that parenteral administration of iron is impractical, dangerous, and unnecessary as a therapeutic procedure. After this time point, many different agents were actually developed, and efforts to complex polynuclear iron oxyhydroxide with carbohydrate ligands began in the 1940s. This ultimately led to the development of numerous intravenous iron carbohydrate complexes, of which there are currently 6 approved reference listed drugs on the US market and 2 generic products, basically allowing injectable iron to be administered to patients. It is really important to acknowledge the criticality of the carbohydrate surface ligands, which impact each product's unique pharmacokinetic and pharmacodynamic profile. As this schematic illustrates, with the 6 available reference listed drugs, each has a different carbohydrate ligand, which comprises the surface characteristics. Also important in understanding this class of drugs is the hypothesized metabolic pathway for these agents. After injection of the IV iron carbohydrate complex, there is a small amount of transient labile iron that appears, which is then quickly bound to transferrin. However, a more important part of the pathway with some yet unknowns throughout the process include the interaction with plasma proteins, which form a yet undescribed protein corona. Then there's presumed subsequent uptake, the mechanisms by which are not fully elucidated, into the cell of interest in the mononuclear phagocytic system, the macrophage. After uptake, there's presumption that it does go into the lysosome for biodegradation. However, again, this is not fully elucidated. And after such time that the iron is moved into a pool that would be available for export, again, the presumption here is that this goes through to ferroportin, which then allows it to be bound to serum transferrin for delivery to the final pharmacologic site of action, which is the bone marrow. And here I've put question marks for areas of the metabolic pathway that have not yet been fully elucidated. And that will tie into our discussion about caveats around the IVIVC and PBPK models. As stated previously, parenteral iron complexes are structurally heterogeneous and produce unique pharmacokinetic and pharmacodynamic profiles. These are representative data from a fairly recent study by Garbowski and colleagues, who conducted a systematic PK/PD study with an intense sampling strategy for 3 IV iron products, which are detailed below. As can be seen in the first panel, looking at total serum iron concentrations, which are also a very relevant clinical measurement that is typically used. And the inset, I think, details this even better. There's clearly a difference in the PK profiles related to serum iron. As you sort of transition to what this might mean in terms of pharmacodynamics, you can look at the second panel, which is serum ferritin, which is essentially a surrogate marker for tissue ferritin, which is, of course, the principal iron storage protein. You can see again illustrated quite clearly that these profiles diverge shortly after drug administration and remain quite different from each other to the end of this sampling period, which was 48 hours. I'd now like to transition into the potential analytes that could be exploited for IVRT, and those broadly fall into 2 domains here. Serum iron, which as I mentioned is a very clinically relevant parameter, easily accessible. However, most importantly, serum iron measurement does not distinguish nanoparticle bound iron from endogenous iron. Moreover, the PK profile of serum iron, if measured less than 24 hours, is likely not reflective of the entire metabolic pathway and then distribution to the pharmacologic tissue. Also importantly, and something that I don't believe clinicians are fully cognizant of, as well as potentially scientists in the field, these iron carbohydrate nanomedicines do interfere with clinical iron assays, which I'll describe also in more detail in terms of assays we used in our project. And this ultimately reduces the quantitative robustness. And then again, as I alluded to earlier, short or sparse serum iron sampling may not fully capture what might be a second phase of bioavailable iron after it's handled and processed by the mononuclear phagocytic system. Looking at non-transferrin bound iron, this represents a very small and transient proportion of iron that is present after IV iron administration. The labile species of this more broader termed NTBI are hypothesized to potentially impact the safety profile of these drug products, but do not impact efficacy. Importantly, the NTBI is comprised of multiple dynamic species in equilibrium, and there is not a universally accepted definition for NTBI. Accordingly, NTBI is also very difficult to measure and depends on the assay type employed, and these can be broadly categorized as capture or chelatable assays or redox assay methodologies. So now I'll move into our project, which was entitled an in vitro in vivo correlation model to predict serum non-transferrin bound iron from intravenous iron compounds. The purpose of our project was to identify appropriate assay methodologies for NTBI that were in fact robust both in vitro and in vivo. We further sought to evaluate then the relationships between the in vitro parameters and the system parameters. In this study, we employed 6 IV iron compounds. 4 of these were reference listed drugs. One of them was a generic for ferrocet, sodium ferrogluconate complex, SFGC, which I'll have that abbreviated as such throughout the remainder of the presentation, as well as 1 preclinical agent from GE Global Healthcare. This table shows the collection of redox and chelatable capture assays we studied in this project. All assays were studied at iron concentrations of 0.952 milligrams of iron per ml. This was selected because it was the predicted Cmax of a 40 milligram per kilogram dose in an average male rat, which was the preclinical species we employed for the in vivo modeling. The majority, to our surprise actually, the majority of assays originally identified exhibited profound interference in the presence of the IV iron carbohydrate compounds. However, we were able to identify an assay, an HPLC desferoxamine-based assay, which used a chelatable methodology, and with some minor modifications, we were able to show that this worked well in vitro as well as in vivo. These 2 panels show the labile iron release profiles in saline and also in pooled rat serum. Of note, we did employ 2 pre-incubation times. We studied these before the addition of DFO to confirm that the iron nanoparticles were not in fact releasing iron during the incubation time. And as you can see by the overlay of the 15-minute and 180-minute pre-incubation times, they are essentially superimposed. So the release essentially starts after the desferoxamine is added. This was a unique aspect of this assay, as we were then able to capture what we then deemed as a release, an in vitro release constant. As you can see in saline compared to serum, the concentrations of labile iron were higher. This is particularly notable for infed if you compare saline to serum, noting, of course, that perhaps serum is a perhaps more biorelevant matrix compared to normal saline. In serum, correspondingly, the labile iron concentrations were lower, and as highlighted, the RLD ferrolicet and the generic sodium ferrogluconate complex were slightly different, but this was not statistically significant. Moving on to the preclinical model, the first panel of this slide shows serum NTBI profiles after IV administration of 40 milligram per kilogram in healthy male rats. Again, the profiles of the RLD ferrolicet were similar to SFTC in this profiling. The in vitro data that we previously looked at led to the generation of two principal parameters, the in vitro release constant, which I alluded to, and then also an in vitro Cmax. When we compared these parameters to the in vivo system parameters, that release, the KE and the Cmax, there was no correlation. However, as you can see on the other panel on this slide, a significant correlation was observed between the in vitro release constant compared to the in vivo Cmax, and the R squared, as you can see there, was 0.711. This was determined by the collective research team to be a relatively good fit, but the caveat being it could be driven by Cmax values. So just to sort of tee up this exploratory study and some of the points made about this complex class of drugs, the characterization of labile iron release from these IV iron carbohydrate nanomedicines in this exploratory analysis did not yield a point-to-point IVIVC. As discussed, a correlation was observed between the in vitro release constant and the in vivo Cmax. However, again, as noted in terms of what analytes represent the optimal focus for such a model, the labile iron really does only represent a small transient fraction of the iron that furnishes the pharmacologic effect. And intuitive to this audience, at nano size range, these drug products have very specific and notably complex properties and characteristics, as well as in vivo behavior that has not been fully characterized, which at present makes in vitro and computational models challenging. And sort of just to overall conclude, more research is generally needed to really understand uptake, the biodegradation processes, and ultimate metabolic fate of these complex drugs before developing further efforts around computational, such as PBPK and IVIVC models. Thanks to the audience, and I look forward to the panel discussion.
Thank you, Dr. Martin, for the presentation. Very fascinating work. And our last speaker for the session is Dr. Feng Zhang. Dr. Zhang is the Associate Professor of Molecular Pharmaceutics and Drug Delivery at the University of Texas at Austin. Please join me. Welcome, Dr. Zhang. And we can see your presentation perfectly. Thank you.
Okay. Do you hear me? Okay.
Yes.
Yeah. Okay. Perfect. Well, so I'm going to present the melt extruded dexamethasone ophthalmic implants. We'll discuss the process, the structure, and the in vitro drug release. So here's the dimension of the implants we're making, and then the composition-wise, we're using micronized dexamethasone in a mixture of PLGA. It's 3-to-1 ratio. The PLGA with SN group, that's 3 parts, and with SN group, that's 1 part. And just be aware, you know, this implant is very small. I mean, each implant is about 1.1 milligram. So for this project, if we look at the property of the drug, dexamethasone does not ionize. So solubility is at 90 microgram per ml. It's a high melting point drug. And the form B is what we're using. We're using micronized dexamethasone for the study. When we run the TGA, because we're using melt extrusion process, so we run the TGA, we realize, dexamethasone, once it melts, it will degrade. The good thing is PLGA has a very low glass transition temperature. So we can run the extrusion process around 100 degrees C. At that temperature, dexamethasone is very stable. Now, we realized dexamethasone and PLGA has a very poor miscibility at elevated temperature. Actually, dexamethasone doesn't dissolve in PLGA during the process. One thing we also observed with the implant, the extruded implant has a similar TG as the raw material itself. That means there's very little drug or no drug is dissolved. This observation, to some extent, has been confirmed by a paper published back in 2016 out of France. And this poor interaction between dexamethasone and PLGA at elevated temperature actually is 2-hour advantage during the process. Now, PLGA, as we all know, PLGA tends to hydrolyze. What we observed is water contents play a very critical role. So if you look at, the 0.4%, S to N group doesn't degrade as much if we hold them at 140 degrees C for certain minutes. However, S to N group, 502H, does degrade. PLGA, typical PLGA as received from manufacturer is about 0.2% moisture content. And in our extrusion process, we don't do any special treatment. We use PLGA used as is. And after melt extrusions, we're seeing about 7% drop in the molecular weight. And we regard that, it's quite stable. And this is our manufacturing process. So we use two-step feeding process in order to maintain consistent diameter. With the HACA mini lab for the extrusion process, there is a laser mic to detect the particle size, and there's a custom-made design puller, and we have a real-time monitoring. So let me go to the details of the manufacturing process. Here's a video. So two-step feeding. We use a volumetric feeder, feed into the stuff feeder that comes in with HACA mini lab. Which then goes through the die right here, and die's control, temperature control. And there's a heated guide plate. And there's a laser mic. So we measure the dimension. It gets pulled. And we're able to achieve very consistent dimension control. So this is around over 25 minutes because the size of the dosage form is so small. So within 25 minutes, we can manufacture about 1,500 unit doses. And as you can tell, it's very consistent. If we had feedback control loop, I think this dimension can be even tighter. So that's the implants we're making. If you look at surface, actually, you couldn't really tell dexamethasone crystals. Drug is all embedded in there. And this is a micro CT scan of the implant. So implants has lots of discrete pores. and there's more pores along the outside, less pores in the middle. Okay, so I mentioned about two-step extrusion process before, and here's a detail. So the first step, we do the extrusion to actually no dimension control. We don't go through those small dies, just use regular die coming on the extruder, and we will make the and then so we extrude them, then we mill them to granules, because only granules can feed consistently. Without consistent feeding, you can't achieve consistent diameter. Okay, and then we do second step extrusions. That's where we achieve the dimension control, because those implants has to be fit into the applicator. So dimension is very critical. So two-step feeding process, volumetric, and then goes through the stuffer. And with this combination, we can achieve the dimension control. And our feed rate is at about 10 grams per hour. And that's equivalent about 10,000 units per hour, because each implant is about 1 milligram. And the puller is custom designed. One thing is challenging with dexamethasone implants is it's very brittle. Drug loading is very high. So it's very brittle, that requires special design puller. And we measure residence time distribution of the dexamethasone in the extruder. So the mean residence time is about 33 minutes. And then potency is 100% and about 7% reduction in the number average molecular weight. And this is why early on, all this thermal stability analysis of the cheap PLGA was based on around 30 minutes. We also find out the porosity of the implants correlates very well to the torque. So the higher the torque, the lower the porosity. And we believe those pores are created due to the air entrapment during the extrusion process. So in a summary, because PLGA has low galactic transition temperature, so we could extrude those implants at relatively low temperature to maintain the drug and polymer stability. And we designed a two-step extrusion process, and with a puller, we could achieve very consistent dimension control. Some of the key physical attributes of implants: diameter, pore size, pore distribution, and also surface roughness. And due to the time constraint, I didn't put too much discussion in the surface roughness. Also, we have collected preliminary data to indicate that PLGA is very stable during the extrusion process, and PLGA is all dispersed as crystalline particles in the PLGA matrix. So now let's move on to the dissolution. For the dissolution, there are publications from Allergan, also in the Ozurdex patent, and they were using a normal saline solution to do the dissolution test. And one thing they observed, and we replot the data based on the data in the patent, is in the rabbit model, there's actually very good correlation between the in vivo and the in vitro. They have a similar profile. Drug is all released over about 1.5 months. Now, just be aware, dexamethasone itself is generally prescribed to last for 3 months in patient. So let's look at dissolution profile. Very little drug. So first of all, there's no burst effect, and very little drug released in the first week. Most drug released in the from in between week 1 and week 4. Okay, and again, this dissolution is run in the normal saline, and after that, all the drug is released after 4 weeks. The reason there's very little initial burst is because we believe the drug is all fully coated with PLGA. Okay. Now, even though in the first week there's a lag phase, but actually there's lots of activities going on. You know, drug water is already start permeating. Okay. And if you look, you know, we're about 10% weight gain after 1 week. And what's interesting, I'll come back to the slides. In the first week, the molecular weight actually dropped substantially, number average molecular weight. And there's also significant median pH. So even though no drug release, but there's water already start penetrating and PLGA start to degrade. And most release happened in the following 3 weeks. Okay. And then there's actually very good correlation between the weight loss and drug release. And when we run the GPC analysis, we find out, you know, in the first week, there was substantial molecular weight decrease and pH drop. That's indicating PLGA start to hydrolyze. And here's SEM image. So the first week, visually, we don't see that much difference. And then, you know, they start breaking up in the following 3 weeks. And here's a cross section. After day 7, most of the pores start close off, and then over the time, the whole matrix is completely dissolved or degraded, and drug is released. Now, we have also carried out the dissolution in the phosphate buffer. And in the phosphate buffer, pH 7.4, is follow the similar release pattern, right? Little burst effect, extended period of no drug release, and then accelerated release over 4 weeks. So basically, it's literally extended out the drug release in the normal saline. Now, in the phosphate buffer, release is much slower because you got this buffering effect, and PLGA hydrolysis slowed down significantly at pH 7.4. That's why drug release is being extended out. So from drug release perspective, we observe triphasic release profile. This is due to the unique structure. The drug loading in the dexamethasone implants is very high, at 60%. But because it's an implant, it's not microsphere, it has a very small surface to volume ratio. That's why they can control the drug release. There's no burst effect. And also because PLGA is trapped, not PLGA, dexamethasone is trapped in there as individual particles, rather than being dissolved in the matrix. That also impact the drug release. No solubility, as I discussed earlier. And then burst release is not observed. After initial lag phase, okay, drug is released over about 3 to 4 weeks. And in the initial lag phase, even though there's no drug release, but PLGA undergoes significant hydrolysis. Okay. In summary, dexamethasone release is controlled by the erosion of the implant matrix. The study I presented here is completed by Mark, my graduate student, and Bebe, my postdoc. And I also like to thank FDA for sponsoring this research. It has been a very fun experience working with you guys. And that's the end of my slide.
Thank you, Dr. Zhang, for your presentation.
Yeah.
I think that concludes the presentations for our session, and I would like to invite the 3 speakers to join and turn on your camera. In addition, I have 3 new panel members I'd like to welcome Mr. Herschel Parrish, who is a principal formulator at Teva Pharmaceutics, and Dr. Mingliang Tan, who is a senior pharmacologist from the Division of Quantitative Methods and Modeling, Office of Research and Standards, OTD FDA, as well as Dr. Diane Burgess from the University of Connecticut. Can everyone still hear me well? I'm sorry, I lost my Zoom screen connecting now, but
we can hear you.
Hopefully you can.
Yes,
okay.
we hear you.
Okay.
Yes, we can.
Okay. While my Zoom is reconnecting, I think we can just get started for the panel discussion. Okay.
Yeah, I think you're muted.
And you're mute.
Sorry, I was automatically muted when I was reconnected. Yeah, I was saying like the three talks we have heard today, I feel like each of them really represents a very unique and complex group of complex products. But I think one thing in common, it appears, I think everyone would agree, the conventional IVIVC is challenging in general. So the first, general topic or question I would like to get the panelists' input is first, what is your general understanding or observation with regard to challenges for developing IVIVC? Like what's our current, where we are in terms of the current understanding, and that we can move on to, discussing potential future steps. So maybe Dr. Patton, you can kick off the discussion.
Yeah, thanks, thanks, Jan. As I've shown in my slides, I think there are various things. First of all, it would depend on the site of administration. If you have if you take nanoparticles that you inject intravenously, the models may not be still, I mean, they may not be adequate to model the extravasation at the moment to outside of the systemic circulation, but there's something we can work on. So I think in terms of tools, when you look at building IVIVC, you need to strive to move away from classical IVIVC because of the what you're interested in is the understanding the drug distribution and ultimately the drug concentration at the site of action. So taking nanoparticles, injecting nanoparticles, we know they're going to extravasate from the systemic circulation to the spleen, to the liver. So building mechanistic models which can comprise extravasation and understanding the local concentrations in the tissue is the way forward. Because from those deep depot, the drug can then be released and be moving to the lymph and going back to the systemic circulation. But this is something you can then try to build, and then plugging the in vitro release you measure, or better, examining the in vitro release mechanistically to understand the variables. Is it the volume? Is it the sink? Is it the local concentration of protein? What drives the in vitro release, and can we model it? Can we understand it from first principles? Because if you do this, and if you then integrate the knowledge of the in vitro batch performance into your model, into PBBM, for example, PBPK model, this will facilitate dramatically the in vitro in vivo correlation, because we're not looking at reproducing the entire in vivo environment. We're looking at examining what drives the dissolution from first principles and then integrating this in the model. I think this is the way forward for complex formulations. We've lost Jan.
So yeah, I think that was a very good response. I would also add that we have been able to do these kind of IVIVCs, you know, just with a more simple basis without taking in all these considerations. And it's not always possible to get the sample right from the injection site. And when we do that, we can do that in animal studies, but not always in the human studies. So that does complicate it a bit, but yes, it's very, very interesting. But it does seem that we are able to, you know, use these other tools such as shifting, time shifting and time scaling. But of course, these are mathematical manipulations, but they do seem to work. Just trying to fill in here while Jan is offline.
Yeah, I agree.
Yeah.
It's probably, I mean, there are some easier methods to scale, you know, from in vitro to in vivo. But you know, if you do this time scaling in a certain species, the question is how does that translate to the human physiology? Because the tissues may not be exactly the same. You know, there will always be that question of, you know, if you've established that correlation in a certain species, would that hold for human? And you know, what kind of scaling would we apply? What is, I don't know if you've got experience in that.
Mm. Well, the only thing I do have experience in is in the rabbit model that we've used and got some very good IVIVCs. We found they were much faster than in humans. And then we were able to nail that down to just the temperature difference, the higher temperature in the rabbits. It's about a core temperature of 39, and that is so critical, particularly with the PLGAs. But then we were able to establish that in vitro, and then, you know, get really good correlation that way. But yeah, there've been some cases where there's too big of a difference between the animal species. But another way is also to maybe look at more than one animal species and try to establish based on that as well.
Yeah, thank you, Dr. Pepe and Dr. Burgess. And I apologize, my Zoom is choosing a perfect timing to act up. So I think, I think, you know, in terms of IVIVC, then I think one interesting point Dr. Barton actually mentioned in her presentation is the PD aspect, to, you know, trying to correlate PK/PD, then how that would help potentially facilitate, right, the IVIVC. So Dr. Barton, do you have like additional points to elaborate here?
Sure, I would just echo Xavier's and Diane's comments about the challenges of the tissue concentrations. I think there's two important facets with the IV iron complexes. One, starting with your question, is this whole PD aspect. There are inherent challenges with the PD markers, which is why we selected a preclinical model. We know that in humans, they have spuriously elevated ferritin, which is the principal PD marker, but this is also an acute phase reactant. So there is just an incredibly complex biological milieu we have to navigate and sort of sort out some of these mechanistic questions. The second point I would make kind of goes back to Dr. Kozak and Dr. Chin's initial comments about, you know, these CQAs and the relationship to drug release. For a class like IV iron compounds or iron carbohydrate complexes, we still really don't have a comprehensive handle on the CQAs. So we're challenged in that area fundamentally too. But I certainly appreciate everyone's comments regarding the challenges on the more mechanistic tissue end.
Thank you. So with that, I think I'd like to see if Mr. Parrish from Teva if you have any experience from your company, you can share here.
I don't think I can add more any significant input than what already has been discussed. So we can just take the next question on that.
Sure. I think there was one question from the audience asking, if there is no to minimal systemic absorption, but the drug is locally acting, right? What are the factors to consider when we are developing IVRT or IVIVC? I think one example could be, ophthalmic product as Dr. Zhang presented. So maybe Dr. Zhang, you could share some thoughts in terms of when you started the project, right, and trying to look into drug release. What were the formulation or manufacturing factors you have considered, and how things worked out for you?
Yeah, so just to claim, I'm not experts in that particular topic, but when we're doing literature reviews, we find out, it seems to me, with Ozurdex, dexamethasone implants, early on you could get some systematic exposure in the patient. The implant itself lasts in the patient about 3 months, but we were talking to some medical doctors at Dell Medical School. They were telling us most implants pretty much gone in 1.5 months, but they're still seeing therapeutic benefit over a 3 months period. So that's just some of the observations we gathered with dexamethasone. And for us, for the dissolution testing wise, there was publication by Allergan indicate they were using, and there was one particular study they did because I think the implants sometimes they will break during the administration. So they were concerned about whole implants versus broken implants. How do they impact drug release in vivo? So they were doing a study using a rabbit model, and they found out there's no difference. And in that case, they're also using normal saline to study, just look at in vitro, is there a difference? It turns out there's no real difference. So we have been using normal saline to do lots of testing, but in the phosphate buffer, the behavior is a lot different. That's what lots of study we're doing now are focusing on why they're different, how they're different.
Right. Thank you for that comment. Just to make sure you can still hear me.
Yeah, we hear you.
Okay. Yeah, I think ophthalmic, or locally acting product definitely carries extra challenges. But when there are challenges, that means there are opportunities. So I think this means one area for the simulation and modeling, and I know our DKMM colleagues have been doing lots of internal and external research. So maybe Dr. Tan, you can comment.
Sure, yeah, thanks, Yan. Yeah, let's comment from the previous panelist. I think, yeah, this is actually a very challenging area for developing IVIVC for these complex products, such as ophthalmic product. I would say, first of all, one of the biggest challenges would be the lack of reliable in vivo data, which may be also for another area which we did not cover today, for example, the dermal and also inhalation, etc. As it may be really challenging to measure drug levels in human local tissues, which is often, impractical and unethical. As we know, in order to do the in vitro in vivo correlation, we do need a reliable PK data, right? And then deconvolute those in vivo PK data into in vivo release profile. Then subsequently, we can correlate that deconvoluted in vivo release data with the in vitro release data to establish IVIVC. However, for those locally acting products, for example, eye tissue, it's very limited human eye data available. That's, I would say, is one of the biggest challenges. Second one, I would say, those kind of limited human data might come from different eye tissue. I know, for example, for eye, mainly from aqueous humor, but also sometimes from other tissue. But those data are usually with sparse data nature. So conventional IVIVC approach, as used for oral dosage form, where usually we use plasma or blood PK data to deconvolute it, which may not work anymore. So I would say thirdly, for another big challenge would be for example, for the ophthalmic IVIVC, it's challenging to obtain the biorelevant in vitro dissolution data, which might not be needed, as we discussed earlier this morning and the previous talks, where for the current PSG, for the quality control purpose and for the BE established purpose. However, for the IVIVC, we do need to have some like biorelevant in vitro dissolution data so that we can have a good correlation in vitro versus in vivo, right? But so for that area, also from the in vitro measurement perspective, there's still some challenge. For example, for the eye, we need to mimic the small eye tear film volume. And also there are some like lacrimal fluid drainage, which happened there. And also what about the mechanical stress caused by eye blinking, etc. So there's some challenge. But on the other hand, actually, there exists some opportunity, I think. As we know, we have a relatively rich animal data available. I think a couple of panelists already like mentioned that, which is a very good resource. One strategy would be we may start with animal model to establish IVIVC and then mechanistically extrapolate the IVIVC there to human by considering the likely change in animal between animal and human for the anatomy and also physiology among different species. That would be one of the opportunity for us to consider. And also for the in vitro measurement side, I think we are seeing that there are some new novel method developed. I think in the next session, we will see a couple of presentation which will show us that. So, but overall, I think by combining the new measurement and also mechanistic, for example, PBPK modeling approach, we may get some better understanding on the release mechanism, both in vitro and in vivo, then correlate them together. Yeah, thanks. That's my.
Yeah, thank you, Dr. Tan. Thank you. So with that, I think, one common challenge or like the conventional understanding on IVIVC is most ideally if we have an IVIVC, then that in vitro dissolution testing alone may be, sufficient to be used as a surrogate to replace the in vivo clinical studies for certain purposes. But I think for complex products, right, one thing we realize is due to the complexity, it may not be an easy approach to obtain. Therefore, I think I want to see like the panelists' point of view, like what if we expand our conventional understanding, but then building more mechanistic understanding into the model to really use that to justify or support, let's say, a design space, right, of a dosage form. Then in that way, I think one restriction with the conventional IVIVC in terms of generalization may be, maybe reduce the barrier. So I want to see, like, from your point of view, what do you think is the next step, or what should be the update, to our conventional concept of IVIVC, like how should we adapt to the new tools and the new mechanisms, and how to potentially you think we should consider in terms of support regulatory applications from your perspective, because this is also something new to the agency and we're learning as well.
I can start maybe, Jan, if you want. I think I'm very much in favor of it because it will bring easier translation between in vitro and in vivo, but also across different species. So if you're able to understand mechanistically what drives your in vitro release and what parameters in vivo are likely to affect it, then that brings understanding of how all those methods translate between one another. And ultimately you will be using, maybe you'll be using your in vitro to extract your formulation performance, whether it is a particle size or a descriptor of some kind to define, you know, the size of your form. If you take PLGA microspheres, you know, we mentioned briefly the impact of size and the impact of degradation. You could imagine that you have you could recreate the pores inside your formulation over time and model the drug diffusion, or if it's you switch to an erosion-based mechanism of release, again, this could be modeled mechanistically based on the flow around the particles, and that will bring a lot of knowledge back into your model and enable this translation to happen. The other way to do it is to complexify your tests to make it more clinically relevant or biopredictive, but ultimately you're not going to be able to model everything. So you know, it's probably best to keep the in vitro test as simple as possible, but still tease out the main formulation characteristic and drug characteristics which influence in vitro and in vivo dissolution and release, and then bring this back into your mechanistic model.
I would just offer aligned with Xavier's comments, you know, looking at some cell-based in vitro release testing where at some point you need to restrict the analytes of interest because obviously the mechanistic depth is complicated, but understanding that these drugs are complex and have complex interaction with the biological milieu, bringing it to another level besides, you know, strictly in vitro testing.
Yeah, thank you for those comments. And Dr. Burgess, I think you have had quite a bit of success, right, in terms of exploring IVIVC for various dosage forms, at least using animal models. And I think like Dr. Pepin, Dr. Barton, or other panelists have mentioned, to keep the release mechanism unchanged is one key. But then I think Dr. Burgess, maybe you could comment on, like, when you design an IVRT method, what is the most critical parameters you have in mind? You know, like for instance, the dissolution medias you have been using, some of them are not necessarily relevant, but they still manage to have a fairly good correlation, right? So what are the key? I feel like really the understanding on the release mechanism and formulation characteristics may be more critical in terms of really understand what factor affects what. I have seen some, like, from zero implementation or consideration of formulation to too many, like a kitchen sink type of consideration on everything. So what is the balance? So what's your experience?
Yeah, it's not just really one thing, but it's understanding the mechanisms, as you said, and it's often not wanting to have a very complex media. That's how we approach it, because if you have too complex media to maybe try to exactly mimic the situation in vivo, then that's going to be very difficult, particularly analytically. So just maybe making some change that may be able to simulate something. So for example, if an enzyme is helping with the degradation in vivo, not necessarily to put the enzyme in there, but maybe speed things up in another way, you know, by pH or adjustment or temperature adjustment or some other type of adjustment with surfactant in some cases for, say, a liposome formulation. So it's just another way to get at what is actually happening in vivo. So another artificial way to do that that doesn't compromise the method and doesn't compromise your analysis.
Thank you. Yeah, I think that's really a very critical point. Then actually, I think there's a specific question for Dr. Zhang. Dr. Zhang, there's a question about the torque in hot melt extrusion. It says it can be affected by feed rate, the screw RPM, degree of fill, and etc. I guess the question is trying to maybe further elaborate exactly why you think it's very important.
Yeah, so the most critical impact, the most critical process variable on the torque that can impact the torque actually is the die temperature. So that's what, now all the other variables, right? The feed rate, the screw speed can impact too, but die temperature has the most critical impact. So by changing die temperature, you can really swing the torque in a very high range. That's what we find out. And also, so there's a couple questions about two-step feeding. This is what we have to do, two-step feeding. When we do one-step feeding, when we feed everything into the feeder that came in with a HACA mini lab, because PLGA has a very low TG, the material tends to melt and will cake inside the feeder. That causes feeder lots of variability in the feed rate. So that's why we use two-step feeders, so we're not choking that stuff feeder that came in with lysis, not lysis, the HACA mini lab extruder that much. That's the purpose. So we're not putting too much material in the that stuffer with the extruder. By doing so, reduce the fluctuation in the torque. As I mentioned earlier, if we had a feedback control loop, our diameter control can be even better. So.
Thank you, Dr. Zhang. And coming back to our first presentation, Dr. Pepin. Actually, personally, I really appreciate you choose Exenatide as the example. That product is extremely complicated, and it is great. Your group is already looking at it, really trying to provide additional mechanistic understanding on this tough product from every aspect. So one question from the audience for you here is, do we know anything about the effect of proteins present in subcutaneous interstitial fluid on the release as well as polymer degradation?
So the presence of protein in the subcutaneous tissue has been measured in human. So there's nice papers by Michel et al. Looking at the IF composition in terms of protein, and it's actually very different from the plasma because the protein, the interstitial fluid is an intrafiltration from plasma. So depending on the size of the proteins, the concentration ratio will be different in the interstitial fluid compared to plasma. And also depending on the hydrostatic pressure. So if you're standing or if you're supine, the pressure in your veins is going to be different and the capillary pressure will be different. So that concentration is very different depending on your posture or depending on the flow across the pressure across the arterial capillaries. So that has been measured, and it's important to consider because they may have so as I said, they may have different concentrations, but it's also going to be dynamic. So when you inject subcutaneously a solution with very little viscosity, you basically flush your interstitial fluid away. And so you replace the interstitial fluid with your formulation, but after a while, the proteins are going to diffuse back in. So that concentration of protein locally in the subcutaneous space is going to be very, very different from plasma, but also dynamically changing over time. So it's really important to integrate this in your model, especially if you've got very strong protein binding, because that will influence the pathway for absorption. If you're bound to a protein, you're going to be absorbed through the lymphatic pathway. So that can add a delay for an immediate release formulation to the to your absorption rate. You may also encounter different macrophage or different cells depending on the pathway you get absorbed through. So if you've got a formulation or if you've got a drug which is sensitive to antibodies, you may lose that drug through a lymphatic absorption if you're bound too much to protein or if you get recognized by antibodies. So it's really, I don't know if I've answered that question, but protein concentration is really key, especially for IR formulations, because they drive the pathway of absorption. But also, I would say in extension also for extended release, even though we have flip-flop kinetics that can drive the rate of absorption, and this is driven by your extended release formulation. Once the drug is released, it may still bind to protein, it may still take a different absorption pathway, which may limit its extent of absorption, so that the understanding of what happens after the release is almost as important as the release itself, because it can limit the AUC. We've seen the example with Exenatide. It's not the same AUC between IR and extended release and IV. So there is something happening after the release, which we need to understand and model, because that limits the AUC. So is it because the drug is metabolized? Is it degraded inside the formulation? Is it unreleased? Now, all these questions are valid and need to be answered in vitro and in vivo, and we need to understand where that loss of AUC comes from to be able to model it.
Thank you for that comment. Next, I would like to invite Dr. Barton to maybe comment a bit on, like, based on your research experiences and current experience on developing iron colloidal systems. What do you think, like, we should further research or study really to maybe bring more weight in terms of assessing bioequivalence from in vivo to in vitro?
Mm-hmm. I think it stems on both ends. One is fully establishing the CQAs with contemporary physicochemical characterization techniques. There's still some challenges even around, for example, DLS. And then on the other side, on the in vivo side, the fundamental challenge, and I'm trained initially as a clinician, is really the assay to measure nanoparticle bound iron, to really understand that that is the analyte we're measuring versus, you know, other collections of analytes. And there's been some efforts in that area. So that's, you know, from my impression, where the dearth of the work should be dedicated in the next few years.
Thank you for that feedback. And I think we still have a few minutes, and I'd like to invite Dr. Tan maybe can share their comment in terms of we're highly encouraging generic applicants to consider novel approaches, right, for developing IVIVC or generally using modeling to support generic development. So do you have any general advice for the audience?
Yeah, thanks. Yeah, I think as we just our panel just discussed, that we are facing some unique challenges in this area for the complex product, like in this session, ophthalmic, implant, injectable. I mean, those area, yeah, really some challenge there, but still, I mean, we still have some opportunity, as we already mentioned there. I think from our side, like FDA, we have recently actually invested a lot of resource or have made some effort in this area, trying to improve the better understanding, especially like the mechanistic understanding of the drug release in vitro and also in vivo. So in this area, I think we are trying to really push this area so that we can have a good IVIVC, so that it can be served as an alternative approach or model integrated approach. In order to do that, for example, as we discussed already, like there are some new novel in vitro method. For example, it has been mentioned like pulsatile microdialysis, and also some new approach, adaptive perfusion method, which will be covered in next session, I believe. So that kind of effort will give us some insight on the way of mimicking the in vivo performance, which we cannot measure actually in vivo. But that's one area, I mean, in vitro release, new novel method, and also from the mechanistic modeling perspective, Dr. Pempe gave very good examples over there. And there's some like the advance which has been made recently for long-acting drugs, ophthalmic drug, dermal even, and also inhalation part. We are getting better and better understanding that area. But again, we need to have a further effort from both industry, academia, and the regulatory agency working together to get some advance in this area, so that we can have a better and also accessible, not better, maybe accessible generic on the market. That's something exciting. And in the meantime, yeah, if there's some like for the generic industry side, if you have any idea or any novel approach, I mean, for the under development, during the under development or submission part, I mean, we have some channel, for example, controlled correspondence or pre-ANDA meetings where we can have a very good communication over there. Yeah, thank you.
Thank you, Dr. Tan. Yeah, I just echo your comment. We highly encourage early engagement. And again, I think all the challenges really present new opportunities for us to update our conventional understanding or thinking on how IVIVC could play a role to support regulatory application. And here we are really trying to explore more design-based or mechanistic way to address. And then in this IVIVC or in vitro diffusion testing is not necessary to be a standalone test. Rather, you could consider that as a part of the totality of evidence approach. Therefore, the type of data, the evidence you may need to support your proposal is not the same level as level A, for instance, the conventional IVIVC. So this is something, we like to emphasize and make sure whenever we mention it for supporting complex generic development, the expectation could be different, and we are open to new proposals. So with that, I think we're at time to conclude our session. I'd like to thank all the speakers and panelists again for the wonderful talks and great presentation. We will take a coffee break and resume at 3 p.m. Eastern Time. Thank you, everybody.